The first kind of movement through a membrane that I want to talk about with you is something known as passive transport. When we look at passive transport, many of you may know this term diffusion. So this is essentially just the spreading out of particles. And what particles want to do is actually diffuse or move down a concentration gradient. In simple terms, they want to move from the area where they're very concentrated to an area where they're not concentrated until they essentially evenly distribute. Okay? So if you plug in an air freshener and you leave the room and you come back in, those molecules from the air freshener are very concentrated near where you plugged in the air freshener, but you may be on the other side of the room and still be able to smell it. That's the idea of this diffusion or movement of particles until they're evenly distributed. And it doesn't have to just be in an aqueous solution. It can be in the air as well. So with passive transport, the passive part of this is that it requires no energy. This is great for the cell. The more it can move passively where it's not going to need energy, the better. And so, but we have to keep in mind that this is only going to occur down the concentration gradient. This means that whatever the cell wants or needs to do has to move with the gradient. So in this case, these pores represent the selectively permeable membrane of the cell. And with that, we look at the random movement of these molecules and eventually this diffusion across this membrane until you reach the same number on one side as the other. Now, in a natural biological system, this is going to be relatively hard to maintain, but they're going to try really, really hard to do it. But there's never just one thing trying to move. So we've got the green trying to move to the right, the purple trying to move to the left, and eventually they're going to bounce around and bump into each other with lots and lots of movement. You have to keep in mind that a cell is a fluid or a moving environment. Always moving. Nothing ever sits still. So back and forth until we get the purple, 3 purples on this side, 3 purples on this side, 6 greens on this side, 6 greens on this side. So they're going to keep this up until they can maintain this over the long haul. Diffusion of water by the process known as osmosis is probably the most common diffusion that we see in our cells. We are always moving water in and out of the cell, and if we couldn't do it by diffusion, so it would no energy involved, it's not going to cost the cell anything, this is critical, okay? We have to be sort of careful with osmosis, though. Osmosis is typically very, very confusing for students, and this is why. So, if I have a setup like this, semi-permeable membrane or selectively permeable membrane in place, the water molecules in this case are going to try to move down the water concentration gradient. So where is water greater? Where are there more water molecules, on the left or on the right? There's more water on the left. Why? Because there are fewer purple solutes. So less purple equals more water in this case. And on this side, on the right, more purple equals less water molecules. You have to think about water as a molecule, don't forget. Okay? So, and as it's trying to move through, you see our water's trying to squeeze through the selectively permeable membrane. And what we want to do is move that water until we actually see this level increase. Why are we going to get more water on the right? Because water is moving down the concentration gradient. It's moving from the left where water is high to the right where water is low. The reason this is often confusing for students is because you tend to focus on the solute. Don't focus on the purple dots. think about the actual concentration of the water. And I know it's very hard to think about water as a concentration. So when we look at the ability of a solution to gain or lose water, we're actually focusing on a term known as tonicity. So what we want to know is, what's the concentration of the solute? What's the concentration of the water? If I know this, I can actually set up a process known as osmoregulation, which is a process that every organism goes through, every cell goes through, to deal with changes in water. So in this case, it's regulating water. So how does a cell regulate water? Depending on the solution that the cell is in. So if a cell is in your body right now, and we want to look at that animal cell. That animal cell, we say, is sitting in either an isotonic, hypotonic, or hypertonic solution. Iso is equal. Hypotonic, hypo means lower, and hyper means higher. So what we want to know is, if you have a cell that is sitting in a solution that is the same concentration as the cell, same concentration of solute as the cell, what's going to happen to the cell? Nothing. Cell volume stays the same. What if I have it in a hypotonic solution? So this means that the solute concentration inside the cell is greater than outside. Which way is water going to want to move? The solute concentration is high inside and low outside. Which way is this water going to move? Water wants to move in. It's the lower concentration of water is inside the cell. Water moves in, and we actually get a cell that may expand and burst or blow up if too much water moves in. What about a hypertonic solution? Solute is relatively low inside, relatively high outside. Water moves which way? Water moves out. So what happens to my cell? It shrinks. I'll show you these in a prettier environment in just a second, but we have to distinguish between plant cells and animal cells here, because plants have a cell wall, right? If they have a cell wall, we have to consider that in this entire process, because the plasma membrane is what's allowing the water through, not the cell wall. The cell wall is all structural. There's no regulation or anything going on with the cell wall. So if we have animal cells at the top, plant cells at the bottom, hypotonic, so low concentration outside, equal concentration, and then high concentration outside of the cell. With our hypertonic solution, what's going to happen? We said with our hypertonic, animal cells will shrivel. Hypotonic, they lyse or explode. But with our plant cells down here at the bottom, plant cells will do the same thing. They will take in water, but they don't explode. Why not? The cell wall is actually going to help maintain what we call a turgid plant cell. The plant cells actually prefer to be this way. It's going to push the chloroplast to the outside for better photosynthesis, and this is really important. The plasma membrane is attached to the cell wall. We saw that when we talked about the plasmodesmata, those connections between two animal cells. And in this case, the cell will shrivel, but it doesn't shrivel quite as far. It actually goes through a process known as plasmolysis, which equals the shrinking of the cell membrane or the shriveling of the cell membrane. Please be very careful with this. Lysis here does not mean break, like it did up here. Okay, so we're breaking connections between the plasma membrane and the cell wall. You see that here, but it doesn't completely break. It stays attached. So it's not like an explosion of the cell. I'm sorry, it's kind of a confusing term for folks, so be careful with that. So if we put a plant cell in a hypertonic solution, we said that the hypertonic solution is going to what? Cause the plasma membrane to shrivel. Does it shrivel? Darn right it does. See all these little squeezed together plasma membranes and inner parts of these plant cells? The outer walls are here. This is the cell wall of the plant cell. But the plasma membrane shrinks down, pushes everything to the middle. This is not a good condition for the plant cell, of course.